So today I'm going to be talking about transcription in prokaryotes. So there's three stages: initiation, elongation, and termination. And transcription is the process of transcribing RNA from DNA. So initiation begins the transcription, and we have our promoter region. And right here is our +1 start, where RNA transcription actually happens. So anything behind the +1 start is considered downstream, and anything this way is considered upstream. So downstream we have our -35 and our -10 consensus sequences, which are considered the most commonly encountered nucleotides. So then we have our RNA transcript, which is going to go 5' to 3'. We see our template strand, which runs 3' to 5' and is what's being transcribed. And then we have our non-template strand. All right, so our core RNA polymerase engages with our sigma factor to give us our hollow enzyme, which starts the promoter region. However, for us to actually start transcribing, we have to take our 3-phosphate RNTP and cleave 2 of those phosphates and take out our sigma factor in order to actually begin adding nucleotides to our 3' end. So this shows you like the end of initiation, beginning of elongation, where our RNA polymerase gets away from our promoter region and starts adding those nucleotides to the 3' end. So elongation, the RNA polymerase changes its shape, keeps moving further and further away from the promoter region, which adds more and more nucleotides to our 3' end. And you see right here, our 5' end stays the same, and the direction of synthesis also stays the same. So termination is how transcription ends. So when the polymerase reaches the terminator site, it actually codes the termination and then stops. You can be RHO dependent termination, which means that you need the RHO factor to catch up to your polymerase, which stops at the termination. So when that catches up, that's what signals the termination. However, if you're RHO independent, this means that you have inverted repeats and then 6 adenines, which when transcribed gives you your RNA DNA pairs, and then 6 uracils, then signaling termination. Regardless of whether you're RHO dependent or independent, you're going to end up with a transcribed RNA that starts at the +1 area and then ends after the terminator. So let's say this gene codes for an enzyme used to produce phenylalanine, but we add 5 base pairs in between our -35 and -10 consensus. So what's going to happen is our RNA polymerase is either not going to be able to transcribe this, or it is, but it's going to have this mutation. So this leads to a deficiency in phenylalanine and can cause intellectual disabilities and physical impairments and conditions. So I ended up using Apple earbuds to describe this phenomenon using the piece that moves as our RNA polymerase. So as our RNA polymerase moves down and unwinds our earbuds, aka our DNA sequence, this is when our RNA begins to get transcribed. And then eventually it's going to hit the terminator site and stop transcription, giving us our 5' to 3' transcribed.